So, hello, hello and welcome to everybody to the national final of FameLab Switzerland. I'm happy that we have people in the audience, and we know we also have a live stream audience, so also welcome to those watching from remote. So, before I say a lot about FameLab, I would like to quickly give the word to Sophia Michael from the British Embassy, who is also going to welcome you, and then I take over again.
Thank you so much. So, good evening, ladies and gentlemen, and it's a fantastic pleasure to welcome you all again to the FameLab Switzerland Final, bringing together talented young science communicators from all across Switzerland every year. Congratulations to all the finalists on making it this far, and I certainly wish you the very best of luck for your performances tonight. I hope you have more luck with the performances than we're having with the weather so far. I would also very much like to thank the organisers, our excellent jury, and all the partners of FameLab Switzerland for organising this excellent competition and for their continued cooperation with Cheltenham Festivals in the UK. FameLab is a great example of international partnership between the UK and Switzerland, and it's one that has established itself firmly in Switzerland over the years thanks to your efforts and enthusiasm. Science communication has an important role to play in our world. There's a great need for clear, accessible science to inform both policy development by governments and also the public debate on the common challenges that we face. And there are strong links between research institutions and universities already, and both the UK and Switzerland are known for their scientific excellence. Initiatives such as FameLab help us to continue this relationship and support its growth by fostering links between the UK and Swiss science communication communities. Our science and technology ambition depends on a strong and varied network of international science and technology partnerships, and Switzerland is one of our most important partners in the UK. To this end, we are supporting various bilateral collaboration projects ranging from fellowships, innovation funding calls, to dedicated exploratory missions in various fields, and I'd be more than happy to talk to you about all the UK and Swiss initiatives in these fields. So, with that, I wish everyone a fantastic evening.
Okay, I rudely didn't introduce myself, so I'm the host of the evening. My name is Caroline Sommer-Trembo, and I'm currently a postdoctoral researcher at the University of Basel and Zurich, and also a passionate science communicator. And I've been involved with FameLab since several years from different angles, also starting with being a participant myself. Especially when I entered this building today, I must say it was such a different feeling when I had my final, because that was in the middle of the COVID pandemic, and basically the audience was restricted to the other candidates, which was a bit sad. And so that's much nicer, because now we're back to normal, and that gives us, of course, the possibility to have an active and personal communication between our participants and the audience. To explain to you a little bit the framework and the rules of FameLab, it is not just a regular local science slam, as we already heard. It's an international network with over 35 different participating countries and 9,000 involved scientists. Also, we have very special rules. For example, the participants are kind of on their own. They don't have slides or something to support their talk. They are allowed props, but basically all the attention is on them. The time restrictions are very strict in FameLab, more than in other science outreach formats. They only have 3 minutes to convince you of their favorite scientific topics. Speaking of the 3 minutes, during these 3 minutes, our jury is going to evaluate our candidates for the so-called 3 Cs. The 3 Cs stand for content, clarity, and charisma. And you will see we have such different personalities today on stage, and they all have their different interpretations of how you bring these 3 Cs on stage, which always makes this event very fun and integrative. I was talking about our jury, so let's quickly introduce our 3 jury members. I'd like to start with Fanny Georgi. She loves in the middle. She loves science, and she looked at science and the scientific communities from very different perspectives. She studied and did her PhD in molecular virology at the University of Heidelberg, but already by then she got curious about how the scientific community worked behind the curtains, so she engaged herself already at that time in university politics. In 2020, she was a founding member of the Swiss Young Academy and later joined the Dean's Office at the UZH, University of Zurich. There's something in common there. And one of her main tasks was the recruitment of new professors, which I find a very exciting one. Now she is at the Swiss National Science Foundation, important for us researchers who want to have their research funded in Switzerland. And she's always happy to see things from new perspectives, and shows she's very much looking forward to this evening with FameLab. Welcome and thank you for taking the time. Please a little applause for Fanny Georgi. Our second member of the jury, Martin Gilbert, is the British Council Director for Switzerland. It's like people waving back, that's nice. The British Council was founded 90 years ago to create a friendly understanding and knowledge between people of the UK and, let's say, the wider world. It works in over 100 countries and connects millions of people through programs and services in English language, arts, education and societies, all topics that he personally is also involved with. The British Council has and is an important factor in the history of FameLab because between 2007 and 2021, the British Council was an important international partner for FameLab, working with the Cheltenham festivals who produced and created FameLab. I'm very happy that the connection between FameLab and the British Council is still so active, for example, by having you here tonight. Martin, of course, he knows FameLab very well, and he's looking forward to tonight's finalists and wishes everybody the best of luck. Welcome, and thank you for being here, Martin. And last but not least, our jury is completed by Thomas Michaels. He's a biophysicist and passionate science communicator. So it's both perspective you're bringing within this event, which is very nice. At the moment, he is an associate professor for soft and living matters at, It's of course, my dad hopefully doesn't see that. Soft and living matter physics at the ETH Zurich. His working group focuses on unraveling the biophysical mechanisms underlying neurodegenerative diseases, such as, for example, Alzheimer and Parkinson. He holds degrees in physics and maths from the ETH and did his PhD in Cambridge. Perfect. Before joining the ETH, the list goes on with the prestigious institutions who joined, for example, the University College London and the Harvard University. We're very happy to have such a specialist for so many fields of research here tonight. Thank you for being here. Speaking of the jury, they're not the only one who can vote for their favorite contribution tonight. This is only after the last participant gave their talk through. I will announce that later, but you can already have in mind your favorite participant here and you can vote for them later. Also, the people on the live stream can use that same QR code later in the break to vote. Of course, the stars of the evenings are our participants. So let's quickly jump over to them. Our first participant today is Sivaniya. She's a postdoctoral researcher at Lausanne University Hospital and is in the field of neuroscience. She has a keen interest for the understanding of differences in how men and women age, with a particular focus on how risk and protective factors shape our aging experiences. So it's basically a topic relevant for all of us since we're basically aging while sitting here. So I'm very happy that we have these science communicators like Sivaniya who tell us about how we should best age, and she's very passionate to communicate this to a broader audience. She's not a newbie in the field, so please welcome. It's all your stage, Sivaniya.
As a child, I was extremely shy and often found solace in books. One of my favorite authors, Frank McCourt, wrote in Angela's Ashes, "You might be poor, your shoes might be broken, but your mind is a palace." As a young immigrant from Sri Lanka, I connected deeply with this message from an Irish teacher who taught me that even in tough times, our minds are powerful sanctuaries. I'm sure you've experienced reading a thrilling book, eager to see how it connects with your life story. Imagine being so engrossed in a story, but as you turn the pages, you find some are torn or missing. Now imagine this book is your life story. Each missing page is a lost memory. This is the reality for millions of people living with Alzheimer's disease, a condition where the memories that shape our stories are no longer accessible. In Alzheimer's, harmful proteins, known as plaques, accumulate in the brain, gradually destroying our memories. Now, if I gave this book to you in another language, say my native language of Tamil, would you notice if a page was missing? Our understanding of Alzheimer's is similarly limited. Most research focuses on white, highly educated individuals, limiting our understanding of how the disease affects diverse individuals. The mind may be a palace, but its doors are often closed by the lack of inclusive practices for gathering knowledge. In my research, I used the UK Biobank, the largest brain imaging data set in the world, with over 500,000 participants living in the United Kingdom. 97% of these participants are white Caucasian, leaving just 3% to represent the 20% of ethnic minorities that belong to the UK. This imbalance is found in several brain imaging data sets. Yet, when we explore diverse individuals in brain health, the results are astonishing. One recent study tracked 255 Black Americans over 17 years to explore the impact of racial discrimination on brain health. It found that greater chronic racial stress was linked to more of these protein buildups linked to Alzheimer's disease. Black Americans are twice as likely as white Americans to develop Alzheimer's, yet are largely underrepresented in our research. Just as a missing page in a book disrupts our story, missing representation in research leaves gaps in our understanding. It's like a palace where only the most privileged are granted entry, while others are locked out. To truly tackle Alzheimer's, we must embrace greater diversity in our research so that we can work towards building a mind that is a palace for everyone.
Thank you very much for this interesting and illustrative presentation. After each candidate, our jury will have directly the possibility and the duty to ask questions. I would say we have an absolute expert in the field as well, so good luck and much fun.
Yeah, of course, I love the topic. So what measures do you think one could do to reduce this imbalance between the representations?
So if I'm understanding correctly, what should we do to reduce this imbalance?
Yes.
Yeah, that's a great question. It's something I think a lot about because it's something I'm quite passionate about. I think one of the biggest issues actually is that there's a lot of mistrust towards scientists. So if you think about, for example, the Tuskegee study that was done in Alabama, these were over 300 Black Americans that were involuntarily taken for research by the government, and they were tracked for syphilis. So researchers knew that they had syphilis, but they didn't do anything about it. And for example, so the study started in 1932. And then in 1947, the medication for syphilis came, but they didn't even give it to them, right? So first they didn't give consent, they didn't get consent from the participants, but then also they intentionally harmed them just for the name of science. So I think there's a lot of mistrust towards scientists and research, and so one of the things I think that we should do is participate in events like this so that we can engage with the public and try to garner greater trust with participants and research and people from minority and racial groups. Yeah.
So I can go on. Thank you very much. It was really interesting, and I think just seeing all those colorful scientists, that's already a measure to diversify science and reduce the mistrust. I'd actually like to go back to the reasons and causes of Alzheimer's. So you mentioned the study that
Yes.
showed that stress causes, or is a risk factor of Alzheimer. Is it stress in general?
Yes,
Do we
so
know that already?
that's a really great question. So the research was done by Simons and colleagues. It was published just this year. And what they did is they administered participants this questionnaire that asked them about experiences of discrimination. So whether they felt they were excluded for certain things, if they experienced receiving racial slurs, for example. And so it was a sort of accumulation of all these things at midlife that they found attributed to greater levels of these harmful proteins. And now, so the question is like, why, right? And so one thing that the researchers posited is that actually, greater stress acts as a mechanism for this greater pathology related to Alzheimer's. So as we know, there's also other research independent of racial discrimination that shows that individuals with who experience greater stress have brain regions that are specifically implicated in Alzheimer's that show reduced volume with time. So it's not just racial discrimination, but for example, other factors like lower socioeconomic status that can contribute to greater stress and also increase your risk for Alzheimer's disease. And again, individuals with lower socioeconomic status aren't also really represented in our research as well. So stress is a really major contributor, but I think there could also be other factors such as, if you're constantly experiencing discrimination, that could affect your vascular pathways as well, increase your hypertension, and just work through the mechanisms of the cardiovascular system to increase risk for Alzheimer's.
Great. So over to me. Now I'll out myself. I'm the one in the jury who knows nothing about science. So I think you've been very successful in actually giving me a greater understanding of an issue that I really hadn't been aware of before. But it's such an important, really, really important issue. This sort of missing representation of whole groups of people.
Yep.
In the health, in this case, in terms of the issues that you were talking about. So I think it's very successful, and I think your use of props was absolutely excellent, and it really helped me to get a picture of the story. So you got your message across to me.
Thank you.
Can I ask you, how did you get the idea of? Did you think long and hard about using the book? You could have used other props.
Yeah, that's a good. Yeah, I mean, Frank McCourt really was someone who was pivotal when I was a child, and this quote it really inspired me, especially since I grew up with similar sort of circumstances as him. He was, grew up in really tough, social circumstances. I understand how difficult it is to kind of make it in a sense, and so I tried really hard to integrate him as well because he meant a lot to me. And then books always meant so much to me, right? And so even if I thought, for example, like I don't know if everyone's watched Sherlock Holmes, but there's the whole like Mind Palace as well. So I really wanted to integrate like books because it means so much to me. And Alzheimer's is all about our memory. Our memory it basically forms who we are, right? And so we learn so much about other people through the books that we read as well. So it's not just our lives, but also other lives. So I thought it was actually perfect that the book is a memoir. It's an autobiography of someone, and Alzheimer's, actually our autobiography, is affected. So I felt like everything worked really nicely, but it started with this quote that really took me through many years. Yeah. Thank you so much for your time.
Our next candidate is Gabriela, a Brazilian physicist dedicated to exploring the enigmas of dark matter, neutrinos, and the origin of matter in the universe. Throughout her career, she worked in several working groups across the globe, and for her PhD, she won the Swiss Society Particle Physics Prize. Now she's a researcher at the University of Zurich, where she plays a leading role in development of an innovative particle detector with applications in physics and nuclear safeguarding. She's a pro in academic storytelling and sees FameLab as another great opportunity of telling another nice story of an interesting topic in astrophysics. I'm very much looking forward to the topic neutrinos, the tiny particles that make physicists go bananas.
Thank you. So, if I may, I just put this a bit closer in the middle. Yeah. Neutrinos are the coolest particles you've probably never heard of. Wait, aren't they inside atoms along with protons? And no, that's neutrons. Neutrinos are much lighter. To compare, if a neutron had the weight of a watermelon, a neutrino would weigh much less than a single grain of salt. Right now, trillions of neutrinos are crossing your body every second. Without stopping or crashing, they fly through as if you were nothing. Nothing, I mean, you're made of atoms. But if an atom was 1,000,000 times the size of this room, its nucleus would be smaller than an apple. So neutrinos can easily cross all this empty space. These swarm of neutrinos come from changes in atomic nuclei. For instance, from radioactivity, after nuclei break apart, or during nuclear fusions. For instance, in the sun, neutrinos are made in the reactions that power the sun. And this image here wasn't captured with light, but captured by capturing neutrinos. This allows us to see not only the surface of the sun, but deep inside the core. And we can also use neutrinos to look inside nuclear reactors, as they produce zillions of neutrinos. Now, a less obvious factor of neutrinos, they are tropical, sweet, and perfect for a snack. Yes, bananas. The potassium in bananas is a tiny bit radioactive, so hundreds of neutrinos are coming from it just now, and from that banana in your backpack. So, neutrinos are everywhere, carrying information from deep inside the sun, nuclear reactors, or even from the explosion of stars, acting as messengers from space. So no wonder we physicists are crazy about neutrinos. And we want to learn more about them. But we still don't even understand how neutrinos have such tiny masses or what type of particles exactly they are. These puzzles keep physicists awake at night. And a famous story is of Ettore Majorana. He proposed a theory about neutrinos in 1937, but then he mysteriously disappeared. Maybe working on neutrinos made him go crazy? Anyway, we're still trying to solve these mysteries. And here goes a last surprising source of neutrinos. You. Yes, you produce thousands of neutrinos every second. Where does that come from? Well, I don't know about you, but I get my source of neutrinos and nutrients from bananas.
I'll start.
I love all these presentations because I'm like, we're all learning so much about each other's like fields of research. So you've finished your first bite
Yeah,
of banana.
the bite is done.
So when it takes so long from having a first theory of something and then proving it, do you think you'll, in your lifetime, will experience solving everything about neutrinos?
That's a very interesting question. So, in fact, neutrinos, there were lots of predictions, and for every single prediction, we managed to prove it. It took many years, like from even saying like they exist to actually detecting them. So, for instance, that was 1937 with Majorana, but actually detecting neutrinos took decades. And then we learned that maybe they have mass, and actually proving that took more decades. And actually, these things take so long, but they are so important in particle physics that already 8 physicists have won Nobel Prizes in physics for things related to neutrinos, because they're really like key particles, but very elusive. As I said, they're so small, so light, so it takes us lots of effort. So your question is, will I manage in my lifetime knowing everything? No, maybe not, but that's also fun for the next generation of physicists who will also have new questions about them. So I think that's probably also fine.
That was really, really interesting. So I got the message that neutrinos are very, very tiny.
Yeah.
Is there anything tinier?
So mathematically, they are point-like because they are elementary particles. But what does that mean? They have, like, when you talk about size, you can think about some interaction, right? So, for instance, an atom has a size, but most of it, of the size of the set, the comparison was like 1,000,000 times larger than this room. Then the nucleus, where all the mass is, would be an apple, right? So it's most of it is just like empty space, especially for a neutrino, because a neutrino would see more of the nucleus, but not much of the rest. So that's why they are tiny, like because in terms of like this interaction area, what we call cross-section, they're just like, this is empty space for me, I'm just flying through, right? So yeah, they're tiny in this sense.
Okay, great, thanks.
Well, thank you very much. It was really exciting. I have to admit, when you pulled out the watermelon, I was expecting a banana, but I'm glad you eventually got the banana. So I was wondering, so you actually work with neutrinos and detectors. So what is it that you try to address in your work?
Yeah, so as I said, it allows us to see deep inside like very dense objects, right? So for instance, we can see neutrinos coming just now from really the core of the sun. Because when you got like photons from the sun, these photons are surface photons, right? And actually they had been in the core of the sun maybe like 100,000 of years ago. So if you want to know what's happening in the sun just now, then you look at neutrinos. But also, you can do this with nuclear reactors, right? And if you do this, you can also prove that the amount of material, like fissile material for nuclear bombs, like plutonium, is really what these nuclear reactors say they have. And this is important for safeguarding purposes. So for instance, you can kind of remotely know and confirm that nuclear reactors are producing the amount of fissile material they say they do. And this is one of the concepts is for safeguarding nuclear reactors, but also you learn a lot of physics if you do that. So it's kind of a double side, like a win-win situation. Yeah.
Okay, thank you again for this very nice talk. Give you a bit of time for the props. Thanks. So either our candidates are not nervous or they're just very, very good in hiding it. I am impressed so far. For now, we stay in the world of physics and the world of atoms. Our next candidate, Shantanu, is our first PhD student of the evening from the field of material sciences and engineering. He is a physicist at the EPFL's Laboratory of Semiconductive Materials, where he focuses on developing novel materials for quantum computing technologies. Science communication is really a passion of his, and he really likes the idea of making complex scientific content easily accessible. To achieve this, he does not only participate in science communication events like this, but he also designs experiments that are both educational and fun for a wider audience. In his free time, he's a very active person. He enjoys running and boxing, which he says helps him to stay focused, but also to energize his work. So I'm looking forward to a very energetic talk on quantum sandwiches, building the computers of tomorrow.
Thank you.
Applause, please.
Once upon a time, there was a king who wanted to reward a wise man. The wise man made a simple request: some grains of rice that can fill all the squares of a chessboard. Isn't it simple? The wise man wanted to place 1 grain of rice in the first square, 2 in the second, and keep on doubling the amount of grain for each subsequent squares. By the time the king reached halfway through the chessboard, he owed all the grains that his entire country could produce. This story shows us how problems that look very simple at the beginning can become very complex. At present, we face a lot of computational challenges, like discovering a new medicine or accurately predicting natural disasters. These problems might appear simple at the beginning, but questions like predicting something or discovering something are really hard for our modern-day computer to solve. They take enormous amount of time and resources. This is where the use of a quantum computer comes in. A quantum computer uses quantum particles, like an electron or a photon, to perform its calculations. These particles are smaller than the size of an atom and exhibit some special properties, like superposition, entanglement, and interference. Because of these special properties, now we can develop a totally new approach to solve these complex problems in an efficient way. So how can we build a quantum computer? Well, one approach will be to capture an electron and try to control its quantum property. We can do this by making a sandwich. But our key ingredient should be atoms of different kinds of semiconducting material. In my sandwich, I have one layer of bread made up of atoms of silicon, which is a semiconductor, and another layer of bread made up of atoms of germanium, which is another kind of semiconductor. And here is my trick. If I can place the layer of germanium exactly on top of my layer of silicon, then we can capture electrons in between the 2 layers of bread. It is like having something really delicious in between the 2 layers of bread. Now we have seen how to capture electrons, but this only solves one part of our puzzle. Building a quantum computer also requires understanding how to control the quantum properties of all these electrons. To conclude, building a quantum computer is a complex task. We are not there yet, but if we succeed, it can unlock enormous potential. And this journey is really interesting. Thank you.
Wow, so fascinating. We go really span such huge topics. And I guess my first question, which you don't have to answer really, is how does a quantum sandwich taste like? I wonder. Anyway, to the serious question. So you mentioned these exponential problems. So can you explain how does a quantum computer manage to solve these complex problems?
Okay, so it's very difficult to make a classical example, but I can try to give one. So assume we are trying to solve a problem, right? And I'm a classical computer. And the problem is go from here to end of the room. And there are 3 ways we can do that. I can either go straight, or take a left turn and go this way, take a right turn and go this way. If I am a classical computer, I will try out all these options one by one, right? But if I am a quantum computer, because of the property of superposition, I have the freedom to split myself into 3 parts. Then I explore all 3 routes simultaneously. It's called parallelism. And the moment I reach the solution, I know this is the fastest part. So this parallelism makes a quantum computer much faster than a classical computer. This is the simplest explanation I can give. It's just a hand-waving approach.
But it's, I understood much more now. One thing that I've always been wondering about new technologies and more computational power is what's the aspect of sustainability? So will quantum computers not only work faster, but also with less energy consumption?
So,
or is it hard to answer? Sorry.
No, it's easier to answer. Like the thing is, a quantum computer is supposed to predict new materials which can contribute towards sustainable energy. So if you want to find out what is the best material to capture carbon dioxide, now we do a lot of simulations. Then we get a bunch of material collection. Then we do test each of their property and figure whether they work out, and it takes a lot of energy. But with quantum computer, we can streamline this process very fast. And then we also expect quantum computer is so powerful, so we will have only 1 or 2 computers working in a country. So the amount of quantum computer that we will use to get the same output of results will be very less. So the energy consumption will be lesser, and then the benefits that you get from a quantum computer can solve the energy problem, environmental problem in a better way.
Okay, so thank you. I think you've succeeded in something that's quite important, that's in enlightening and creating curiosity. So I really can't wait to see what happens in this quantum computing future. Could my mobile phone, which I suppose is a little computer, could that ever be a quantum computer?
So it might happen in a way that you can connect your mobile phone to a quantum computer through quantum internet.
Brilliant.
So you can use it.
Okay, thank you.
Thank you.
Great. Thank you again. And now we will have our next participant, Jose, a PhD student in material sciences at the ETH. He studied nanoscience in Barcelona and physics with a focus on sustainability in Amsterdam before coming here to Switzerland to start his PhD. We all know that Switzerland provides a very nice research environment, but this was not the only driving factor for José to come here, because he grew up in the Pyrenees and spending quite a time in Amsterdam, he simply missed the mountains. What we all know in life is we cannot have it all, because now that he has the mountains, he misses the sun, which is one reason why he wants to talk about the sun in public today. Jumping elephants in the great solar leap. The stage is yours, José.
Think about your journey here today. Did you notice any solar cell? We could produce enough electricity to power Switzerland, covering less than 1% of its territory with solar cells, which sounds small, but it's actually around the size of beautiful Lake Geneva. Why so much? Well, because solar cells can only turn around 25% of the energy in sunlight into useful electric energy. If we had perfect solar cells, we would need 4 times less land, which is, okay, let me check with my solar calculator. The area of the city of Zurich. However, we will never have perfect solar cells, no matter what physicists like me do, and because the answer is not in the technology, but on the sun itself. Sun sends us light, light is made out of photon, and all photons have different energies, but solar cells work best with only photons that have the right amount of energy. Let me explain this better, turning those photons into elephantons. We have a very diverse crowd of elephantons here. We have blue, very energetic elephantons. Then we have the green, medium energy elephantons, and we also have some red, very chill elephantons. Now, picture the elephantons playing a jumping game. They need to jump over a hurdle. The red, very chill elephantons cannot jump high enough, so they don't make it. The green elephants have the right amount of energy to clear the hurdle. And the blue, high-energy elephants have so much energy that they jump way above the hurdle. However, since the objective was just to clear it, all this extra energy, it's wasted. And no matter where the hurdle is, there will always be elephants that will miss and elephants that will waste their energy. Solar cells face the same problem with photons from the sun. Some photons lack energy, and some photons waste it. And this is why perfect solar cells won't happen. And therefore, covering Zurich with solar cells will not be enough to power Switzerland. So now the question is, should we give more room to an inefficient technology? Before you answer, let's address the final elephant in the room tonight. And it's the following. I don't know if on your way here you saw solar cells, but I assure you, you saw a lot of room being given to an equally inefficient technology, cars. Cars only turn, while solar cells turn 25% of the sunlight into electricity, cars only turn 25% of the energy in fuel into movement. The rest is wasted heat. And nevertheless, we have covered more than the area of Lake Geneva in Switzerland with infrastructure for cars, roads, and parkings. So next time, think about that. And maybe with all the elephants here, we deserve to give solar cell a bit more room to shine.
Fantastic. When you came on stage, I thought, okay, everybody had something, and it looked like you didn't have anything. But actually, where did you keep all of this?
Well, I read that we would actually not be given time to mount the prompts, so that's why I thought I need to carry them with me.
And well, I was wondering. So you mentioned efficiency, but there is also a question also of materials, right? So I'm not an expert, but I heard that to create these solar cells, I mean, you need very precious metals and so on.
Yeah.
So how does this issue compare, for example, or relative to the efficiency issue?
Yeah. So yes, you do need some silver, for example, but most of a solar cell is silicon, which is a material we have everywhere, and that it's very cheap to produce. That's actually, solar cell, it's solar energy. It's the cheapest kind of energy in most countries in the world already. And what you're talking about, yes, the energy cost of producing solar cells, this can be calculated through what is called the energy payback time, by how long we need to have solar cells producing electricity in order to cover for the energy that went into fabricating them. In Switzerland, it's around a year and a half, while the lifetime of a solar cell, it's over 20 years. So it's really not a debate.
Super interesting. So I read different times for the energy. What was it? Like the time you need to have the solar cell running until you've produced the energy that you took to produce it. Interestingly, comparing it to cars, it took millions of years to generate the fuel that we're using. Isn't it the case?
Exactly. Exactly. And we have photons and electrons everywhere, and we don't have oil everywhere.
The precursors of the oil. But now to my actual question there. Could we use this, heat that we produce on the side in cars? Like we try to be efficient and use the heat in our wastewater? Could we combine solar panels with something else?
Yes. So for example, this is not really my topic, but this number that I gave, 25% of the energy in fossil fuels going into moving the car, that's the case for gasoline and diesel car. Electric cars have efficiencies of 90%. So if you could produce the electricity by solar or renewables and then have an electric car, you'd end up saving a lot of the inefficiencies all the way. And cars do, just for the matter, like I show a Formula 1 car. Those cars have efficiencies of up to 40%, or that's what they claim, among other things because they recycle this waste heat that they have. However, after 100 years of development of cars, they are still commercially at 25%, while solar cells have been developed for way shorter, and they are at the same number. Just another thought.
Yeah, thank you. That was a very clear description for me that there are some sort of sources of energy are much more efficient than other sources of energy, and it makes me think, well, obviously there's a few reasons why the petrochemical industry has managed to persuade us for such a long time that cars are the way forward when fueled by diesel and petrol. Is it because it's only now that the research is being done on things like solar, or has that been around for ages?
Not for ages, but for quite a while. And so, for example, the limit of solar energy and how much we could obtain out of it, this was calculated 50 years ago. So we've known that quite a while. And you know the story that electric cars were more or less at time with combustion cars which were there. So those are more political reasons, I would say, and communication reasons, that they are really scientific reasons. That's the same reason why some people still fringe a bit when they see solar cells on the way, but no one has noticed the cars and the roads around us. We just grown used to it.
Thank you, Jose. While Jose is collecting his elephants, a quick information for the audience and for the jury as well. After the next candidate, we will have a quick break. Please don't stay away for long, but just a quick toilet break of like 5 to 10 minutes before we go on. Our next candidate, Michael Morari, is a truly international candidate. He was born in the US, he grew up in Switzerland, but he's Austrian. His love for animals started with Calvin and Hobbes comics, grew into love for sci-fi with Spider-Man movies, and matured into love of science. And he studied biology and bio and chemical engineering. Currently in a career transition, as I heard. I heard him last year at the Science Slam in Basel, and trust me, he knows how to entertain. I must say, personally, content-wise, I'm very much looking forward, since I do a lot of fieldwork in a very remote place in Africa, where there's, for example, the black mamba, and where you know if you got bitten by that thing, you only have a very few minutes until a very painful death. So please tell us about You Got Bit, learn how to make antivenom in 3 minutes or less. Thank you.
So I brought with me today. Ah! I got bit! And I've only got 2 minutes and 55 seconds to tell you how to make snake antivenom. I'm Mike. Let me tell you what's happening in my body. Snake venom is a cocktail of toxins. Up to 200 different toxins are in my body right now. There's neurotoxins blocking the paths between my nerves and my muscles, paralyzing me. There's myotoxins destroying my muscles. There's metalloproteinases, which are punching holes into my blood vessels. All of this is happening within minutes, and my immune system has no chance. We need something to make antivenom. Or we need someone. Meet Biscuit the Horse! Yes, because we can inject Biscuit the Horse with a tiny drop of venom. Boop. I'm Biscuit the Horse. Let me tell you what's happening in my body. The tiny drop of venom has no chance against my strong horse immune system. It recognizes the surface particles of the toxins and creates antibodies. Those antibodies bind specifically to the toxins and disable them. Yeah, and then I can go in and boop, extract the serum, part of the blood, identify the antibodies in it, purify it, and then I've created the snake antivenom against all snakes. It's not that simple. No, Biscuit the Horse, it's not that simple. We like to have fun up here, but the reality is far more complex. Snake species, each species has a unique blend of cocktail toxins. That means that this antivenom I made is only going to work against this specific species of snake. Also, with the snakes and the horse, it's incredibly expensive. And now imagine, where do snake bites happen? Of the 3 million snake bites that happen every year, the vast majority happen in regions of the world like sub-Saharan Africa or South Asia. in communities that are predominantly rural or low income. And the cost of this antivenom can be prohibitively expensive, if you can even get your hands on one. But there's hope. The World Health Organization has designated this disease as neglected and is funding $80 million over the next 5 years. And there's promising research into alternative lab-grown antibodies that are more selective and more economic. So, does that mean you don't have to inject me anymore? Well, it'll be 10 or 20 years away. Say, how do you know so much about immunology? I've got a PhD in snakes!
Thank you very much. That was very... Are you all right?
It's fine. It's like I was embodying the blood circulation being cut off.
Method acting on stage. Luckily, we have a break afterwards. Theoretically, could you combine lyophilized antibodies against all known toxins and venoms in one super treatment?
Great question. The universal antivenom, is that a thing? Well, it comes up to some limitations in this method by using animal serum. So imagine I've got 10 different species of snakes, and they all bite Little Biscuit the horse here. And then we extract that serum. So what's in that serum? Actually, 90% of the antibodies in that serum is just other antibodies from the horse's immune system, and only 10% is actually against the snake venom. And then, since we have 10 different ones, a tenth of that 10%, so 1%, is against each of those different venoms. Meaning, yes, it's more universal, but you need far more dosage for it to have an effect.
These snakes, how come they've developed such really strong venoms?
Well, it's an evolutionary question, and actually, so the venom glands have evolved, 10 or 20 times separately in evolution, and it has different uses. Some give you an advantage as a hunter to disable prey. Others is more defensive, also in other animals where they use it as defensive poisonous quills, for example. So it's about giving an evolutionary edge.
I loved your energy. I'm still wondering how you're still alive after all of this. But, if you had to give, I guess you cannot make an antivenom in 3 minutes, right? I guess that's the point.
Yeah.
But,
you can explain it in 3 minutes.
Yes, exactly. But what could you do, actually, to, slow down or, at least save yourself as much as possible until a rescue would come, I guess, in maybe the more developed areas?
Yeah, well, what you should not do is kind of those Rambo Boy Scout ideas of, let's cut in with a knife and suck out the snake venom. No, that doesn't work. You also shouldn't make a tourniquet to cut off the blood flow. Basically, you have to get as fast as possible to a place that has the antivenom, and also try to immobilize so your movement and your heart pumping does not spread the toxin as fast as far. But that's actually a big issue, and part of those 80 million in World Health Organization funding, it's not only going to the antivenom research. A lot of it is going into awareness, preventative measures, and also infrastructure to enable, that you have some vehicle to get you fast enough to a hospital.
Thank you, Michael. So we'll have a break of 5 to 10 minutes. You can use the bar, you can use the toilets outside, but I would ask you to be back on time. A larger break will be given after the last talk where you can enjoy more drinks and a more extensive break. Hello, hello. Can I quickly ask everybody to slowly go back to their seats, please? Okay, welcome everybody back from the little break. Our first candidate for the second half of the FameLab is Julia. Close to her hometown in Germany, where she actually grew up on a farm, she did her bachelor in computer sciences. She wanted to see a bit more of the world then and went for a semester to the US, both as a data science student and a tennis athlete. For her master's, she came to the ETH and specialized in theoretical computer science and machine learning, and she's specifically drawn to the algorithms and want to explain to you how algorithms can solve amazing problems in our world. The stable marriage problem, dating advice from a computer scientist. I'm looking forward. Stage is yours.
Today is going to be the first and only time you're going to take dating advice from a computer scientist. So who of you has seen Bridgerton? Anyone? Nice, okay. So imagine the marriage market of the 19th century. It's very traditional, very heteronormative, and also everyone really wants to get married. Let's say we have the same number of men and women. And now this season begins and they check each other out and rank their potential partners by who would be first choice, second choice, and so on. And now the drama begins. But when we computer scientists watch Bridgerton, we wonder, can't we just match them in a stable way so no one breaks up? You can imagine that there could be matchings where two people prefer each other over their matched partners. For example, if we do not match Colin and Penelope, they'll probably find each other and leave their partners and run off together. So this was not a stable matching then. Luckily, we have an algorithm to prevent this, and here's how it goes. All women go to one side, and all men go to the other side. And now men are really confident. They approach their number one and propose. Now, women can't do much in the 19th century. They can just say yes or no. And if she's single, she'll just say yes to anyone and get engaged. But what if she is already engaged? Then she compares her fiancé to the proposing man and just takes the one that she ranked higher. The other man sadly goes back, eliminates her from his ranking, and goes for his new number one next. When everyone is engaged, we can do a mass wedding because we have found a stable matching. Yes. So this is a very, very nice algorithm that is being widely used. Not to marry people, of course, but, for example, to assign 30,000 medical students to hospitals every year, or to match your phone to a server when you surf the internet. Now, what do you think? Who is better off in this algorithm? Is it the man, because they're confident they go for number one and they have the active role? Or is it maybe the women, because they can decline offers and improve? What does your intuition say? Who says it's better for the men? Okay, who says it's better for the women? A mathematical proof gives us a very surprising answer. Every single man gets the best possible stable option, and every single woman gets the worst one. So here's my dating advice. Be active, be confident, and go for your number one. Thank you. Any questions?
Not sure if we should organize dating in that way. But one point you've not touched on is how do you determine the sequence of who can choose first? Because that person is obviously in the best position, no?
Good question. It actually doesn't matter. But this is something you have to prove, and the intuitive reason behind it is that, well, maybe someone goes first, she maybe says yes, but then if the other guy is better, she'll like dump him anyway and go for the better guy. So it doesn't really matter if he asks first, if he will get dumped later on anyway. So the order in which the man asks doesn't matter.
Thanks very much. I loved that that presentation and the comparing sort of this mating ritual with some kind of algorithm that also works for matching up people to medical institutions. It's really interesting, and for me, I've taken the big thing I've learned. It's a bit late for me, of course, but looking back 40 years, being active. If I've understood it, that's the really important thing.
Yes, so I think, of course, you cannot transfer this one-to-one to reality. I do admit we cannot force people into marriage, and I also do admit that maybe I wouldn't just want to marry anyone, because, for example, the women, they just always say yes, apparently having anyone is better than being single. I wouldn't agree with that. But yeah, it's interesting to see that just the active part of this is so much better off.
Very interesting. I think I'm going to try it out to see what happens. But I guess it depends also on the size of the group, right? So is this prediction for the stable equilibrium, is it depending on like a huge sample? Or does it work also with like a finite size of a population?
That's an amazing thing about this algorithm. It always works. So the only thing we need is same number of men and women, and then they just make the lists, and it works. It works perfectly every time. Yes. Thank you.
Thank you, and let's move directly further to Emmanuel, PhD student in microbiology. He started his academic path with a degree in biology from his hometown, while it remains a mystery what this hometown is, but maybe we can talk about this later. He then did a master's in pharmaceutical biotechnology in Bologna and enhanced his research skill at Barcelona Center for Genomic Regulation. He is currently advancing his PhD in microbiology and immunology at the ETH, and he's very passionate about bridging the gap between scientific research and public understanding. And as we can see, he's more than well prepared in terms of prop and explaining to us the endless battle between bacteria and phages.
Hi everyone, I'm a friendly bacterial cell, and I'm very happy to be here in this cozy place. Ouch, are you also getting bitten by something? Oh no, that's a phage, a virus that can infect bacteria like us. Now this sneaky phage has injected its DNA, that's a problem. It's injected its DNA inside me, it's trying to hijack my body. But you know what? I'm not defenseless. I have something on my DNA that to try to handle this. Let's see if I can find it here. It's a little bit broken, but it still works. I'm pretty sure about that. Here it is. It's my antiphage Hermie knife. So now I can see this is foreign DNA because I wisely decorated my DNA with all these methyl groups while this invader didn't bother. So I can use my restriction modification tool to cut the phage DNA, and then problem solved. But wait, another phage attacks, and this one was a bit smarter because it got its DNA decorated too. Still looks suspicious to me. I think I'm gonna use my favorite tool. It's called CRISPR-Cas. With this tool, I can precisely cut the phage DNA here, and the genius part is that I can save a snippet of the DNA into my DNA. So next time this phage attacks me, I will immediately remember and destroy it. Now, CRISPR-Cas is not only cool for me. Human scientists have discovered its potential over a decade ago, and now CRISPR technology it offers the possibility to modify genes in our DNA and cure genetic diseases. Just last year, the first CRISPR therapy was approved to cure sickle cell disease, a one-time treatment that can solve the problem for life. Not bad for a bacterial defense tool, huh? But back to my story, I get attacked again by another phage. This time it's a bit more successful. And this phage is actually very tricky. It's got an anti-CRISPR protein that can block, here, where is my CRISPR? Can block by CRISPR tool, so I cannot use it anymore. So now, what can I do? Now I'm in serious trouble here, okay? But you know what? You know what? I don't want to let the phage spread to you, my fellow bacterial friends. So I'm going to use my last resort tool. It's called abortive infection. I will sacrifice myself so that you can all can be safe from the phage. Bye. Well, it's a complicated tool here, huh? I knew it was going to happen.
Wow, amazing. I think you should try to sell this as a Swiss Army knife or so. It's like all the different tools that you have there. It's really amazing. And I loved how you continue despite the collapse behind your back. So well done on that. So first question, which is your hometown?
Say it again.
Your hometown, which one is it?
So yeah, I'm from Cagliari, Sardinia, in the island in Italy. It's basically the ball that the boat is kicking.
Very nice. And you mentioned about this treatment for sickle cell anemia. Can you explain very quickly how it works?
So sickle cell disease, it's a genetic disorder. So it's a point mutation in your cell, and basically with this CRISPR, I didn't explain the details, but you can really, if you know the sequence of the gene that is mutated and causes the disease, you can really go and target and modify it. So in this case, what you can do is you take this blood from the patient that you modify with CRISPR, and then you can transfuse back, and then this disease is really virtually disappears because it's corrected by the tool.
Super cool, and I finally know what I'll do with like the hundreds of meeting bands that I have at home. Use them for science communication. So based on your title, I was actually wondering beforehand if you are also working with phages themselves as treatment. Because, I mean, phage therapy has been around for ages.
So it's not really the topic of my group. This is more a bit of my passion. So I work with bacteria and phages, but it's more of an ecological way, you know, how they interact and how the phage, the killing bacteria, how this can affect the bacterial population. Of course, phage therapy is now getting it's becoming a hot topic because of the coming antimicrobial resistant crisis. So it's definitely a topic that I'm really keen and would like to explore maybe afterwards.
But as expert in phages and bacteria, what could be limitations to the usage of phage
phages,
therapy?
of course, are viruses that can kill bacteria. So if you have a bacteria that is a pathogen and is causing disease to humans, then you can either use antibiotics, which is now what the go-to treatment, especially in the Western world. but then we have seen the bacteria are becoming more and more resistant to it. So that's the idea of, which is very old, it's at least from the 50s or 60s, to use phages, which are viruses that can kill specific bacteria. The thing is that I try to explain here is that, I mean, even with phages, bacteria, they still develop resistance to it. So it's an interesting study showed that if you combine antibiotics and a phage, the target to the bacteria, then the bacteria sometimes has to decide, you know, do I want to be resistant to the virus or to the antibiotics? And this using combination of these two, it's probably going to be helpful treatment. So there is definitely some room for phage therapy to help the antibiotic crisis.
That was very interesting, and I really learned something. I've managed to go through life so far without never hearing the word phage before. So it was a real eye-opener to me that there's this battle going on between the phages and the bacteria. Do you personally have a favorite?
Oh, the phage, yeah, I have to say I'm a little bit boring in that sense. I use a very standard bacteria and then a very standard phage, which have been studied since the, I don't know, 50s or 60s. So it's not like very original, but yeah, I do read about other types of phages to understand all the intricacy, because yeah, all these defense mechanisms are actually very specific sometimes to some species. So yeah, I would say I very like lambda as a very standard phage. It's the most famous one. And yeah, and they also, you know, they inspired. Actually
they're
wanted to
very
have
cute.
different shades, but then it was a bit more complicated to fit the conference strap.
Thank you, that was very creative. Yes. Maybe somebody could, maybe get some help, don't worry about it. Thank you. I already start to introduce our next candidate, Daksha, a PhD student in physics. She's an experimental physicist from India and now dives into the scientific world of Switzerland. We're going to hear about her own work, and next to her work she finds peace in painting and dancing. By the way, I find that very important to have a life besides science. It kept me sane as well. It is important for her to effectively communicate her work with a bigger audience, and I'm very happy that you brought up a topic in your introduction saying you need to overcome stage fear, and that's a real thing because us scientists, we're not trained to be on the stage at all, and everybody who ever attends conferences where us scientists are actually in our comfort zone, you know, speaking about our topics with the scientific community, even there you can see that most of us are really not made for this. So I'd like to have a big applause for everybody overcoming a big stage fear here, and I know what I'm talking about. Each time I'm participating in a science slam, there's these 5 minutes before that I think, why? Why I'm not sitting in front of Netflix and just chilling? But we're all motivated to do that, but it takes quite some boldness to do it. So please, a big applause for everybody, and especially now for Daksha to be on that stage.
I'm sure all of you might have heard about the atom in your school days. It is in fact quite similar to the solar system. Just like the planets move around the sun and spin on their axis, in the atom, electrons move around the nucleus. Though this is not the bad picture of the atom, it is not accurate. Especially electrons, they never spin on their axis. But hold on. Physicists do say that electrons have spin. So what do they mean? Well, spin perhaps is one of the most terribly misleading terms in physics. You see, electrons are like these tiny little magnets similar to those that you stick on your refrigerator. It is this magnetic property that is referred to as spin in case of electrons. Confused yet? Don't worry, it gets weirder. So, electrons don't really spin, but let's rotate them on our own forcefully to see what happens, okay? Let's start with something very simple. Look at this cup, for instance. If I rotate it all the way around in the same direction to a full 360-degree turn, it looks the same as it did before. Agree? Most of the things that are around us behaves in the same way. Let's try to do the same with my belt. So I have a belt whose one end I will fix in my book. Keep an eye on it. Now let us twist one of the ends of my belt to one full turn. Keep an eye on it. This is one full turn, right? The belt is twisted. The question is, can I untwist it back? And you would say, of course, just undo the twist. But this is not what I did with the cup. I rotated it in one direction till the point it comes back to its original position. Can I do the same here? Of course, I can move this free end of the book around the book and do whatnot, because it is not the same as undoing. But you see, it is impossible to bring it back to its original untwisted form. But what if I do 2 full rotations instead of 1? Let's try. So 1 full turn and 2 full turns. Ready? Now watch. Surprise! What if I tell you electrons in the same way also need 2 full rotations instead of 1 to come back to their original position? So while electrons spin and come back to their original position with only 1 turn, electrons play by their own rules. This Dirac bell trick is just one of the many weird wonders of the quantum world. So the next time you're trying to tie your belt in the morning and it seems like it's taking forever, just remember, it could be worse. You could be an electron. Thank you.
I really learned something new again in each of the presentations. It's so fun. Thank you very much.
Yeah.
Question.
Yeah.
Does that mean that electrons are attached to something?
No.
Because the difference was that your belt was attached to the book,
Okay.
but the cup not. No. Why?
So what I'm trying to show here is if you rotate an electron, what will happen? So this is very simplistic model because you need, so in many of the objects you need 1 full rotations to come back, right? But electrons are very different. You need 2 full rotations. And in what we call as classical physics, means what we see in everyday life, it's very difficult to find in the picture of electrons to see an object which needs 2 full rotations. So this is a very simplistic model to tell you that the belt also needs 2 full rotations to come back to its original position. So this is kind of an analogy between the rotation of the electron and something that you can see, something you can visualize. So this was the easiest thing.
Amazing. How did you come up with the idea of the belt?
So it's like Dirac, the physicist, he proposed this idea of electron, how this electron needs 2 rotations, and he explained this idea using this belt. So I thought it's a nice idea of telling it to everyone and understanding by myself as well.
I was actually going to ask the same question because I think it's the analogy is really is very good and makes it sort of understandable to someone like myself. So well done on coming up with a really good way of describing something.
Thank you. Thank you.
Okay, our second last speaker of the evening, Emma, is a PhD student in physics. I have the slight idea that our physics people are maybe a bit better in explaining their science because in all the semi-files there's still a lot of biologists and from other fields, but the final is full of physicists. That's very nice. It means you're doing your job very good. Emma comes from Norway, where she grew up in such a small place that she usually answered the question, "Where are you from?" by naming the nearest fjord or glacier. Her love for physics originated from learning about space, and although astrophysics was her entry point at university, she then went into nuclear and medical physics after a few years. After a couple of stays abroad, she came to Switzerland for her PhD, and now she tells people where she lives by naming the nearest lake or mountain range. Please tell us about hyperpolarization. What is polarization and how is it hyper? Welcome, Emma.
By show of hands from the audience, who remembers the children's book or game of Where's Waldo? Où est Charlie? Okay, great. But it is a game. We have a page with hundreds of people where you're trying to find the one just like this. Now, being a physicist is similar to searching for Waldo. Okay, not all physicists, but for me it is. You see, the way you search for Waldo on a page, I search for a specific type of atom hidden among others. Truth is, I cheat. Before I tell you how, let me explain. As a nuclear physicist, I work with atoms. They are very small, so it's really a detector that measures an electrical signal. I can spend a long time searching for my signal, my Waldo, or I can just create my own Waldo page. So before I reveal my secret, I want to tell you how we actually observe atoms. So indeed, atoms consist of a nucleus and an electron, which we were lucky to hear about just before. And for simplification, I'm going to say that they spin. How they spin, we attribute a value to. Now, a lot of nuclei of interest have a spin value of 1/2. Now, those have two possibilities. They are either spinning positively or opposite negatively, so up or down. Truth is, my detector doesn't see just one nucleus because they come in many, many millions. If you add up all of those millions of up and down pointing nuclei, that's what we call polarization. So indeed, between those two, many of them will cancel each other out, but there will be some excess ones, some extra ones in either of the categories. It's those extra ones that is the reason you have a signal. That's my Waldo. Truth is, some nuclei are eligible for a little bit of intervention. That means in the right environment, a scientist can flip, that is the correct term, spins from one category to the other. That means that now you have a few more extra in one of the categories than you had before. Therefore, the signal is much stronger. So that's my secret. With hyperpolarization, I'm making my Waldo 100 times larger than what he is, and detecting Waldo, as well as my hyperpolarized nuclei, is a lot easier, even if I put him on a different page. Thank you.
So very nice, and I loved the outfit. And we found you, so that's...
I'm here.
Yeah, exactly. So you said you're looking for very special atoms.
Yeah.
Which atoms are you looking for?
So there are certain isotopes that are very suitable for this type of intervention. And in my field, which is medical physics, it's something that you actually can safely introduce into a human and kind of track and follow. So it's certain specific isotopes that are eligible for this intervention and that are of interest when it comes to, for example, metabolism.
So now that we're already introducing those atoms to the human body, I mean, what the the public is often really scared of is creating black holes, and then we're all just gonna, disappear. Is changing the spin of particles and atoms dangerous?
No. So the reason why not is because you're only temporarily changing the state. They are always going to relax back to where they came from. Because nuclei, or atoms, they have a stable state they prefer to be in. And so even if you alter it, it's going to return back to its normal state.
How long do they stay in the,
in this state? So it changes. So the fun thing, which I can't get into, but it's all to do with magnetic fields and temperature. And so if you want to move it from where we're creating this state, which is 1 Kelvin, that's not where the human temperature is, that's at 37 Celsius. So you can keep it cold and in a magnetic field, and it stays for, it can be minutes per hours or something like this before it naturally goes back. But as soon as you change it into, let's say, a biological environment, we're talking between seconds and minutes. So that's, you can say, a disadvantage. But you can also use it for your advantage to, for example, not see recycled signal if you're introducing it into a body. You're sure that that's my signal, and it hasn't gone through multiple processes through the body. So you're taking advantage of what could have been a drawback.
So I've learned the concept of polarization, obviously important, and flipping is something that's also really important. Where did who developed that?
Flipping?
Yeah.
Good question. So this goes back a while. So I'm mentioning the applications in medical physics, but it's also suitable for geophysics, which I'm not going to touch because I don't know much about that. But we're looking at the idea of changing these states decades. And then, especially 2 decades ago, was a huge leap that happened to make it possible to actually introduce it to medical physics, because instrumentation could develop, and so you could, what's important is this rapid change, right? And so a few very key changes to instrumentation made that possible. So it moved from a very stable and cold environment into actually today being in use, FDA approval for certain isotopes in the US, and plenty of clinical trials around Europe as well. So it's quite the escalation. Thank you so much.
Thank you. And we're coming to the last candidate of the evening. Is it okay for you to have the table in the middle? Maybe somebody, because I have back issues, maybe somebody could just... Thank you so much. So our next candidate is Christian. Not a physicist. He's a PhD student at the Swiss Tropical and Public Health Institute. He was born in a village close to Freiburg. I didn't know that, because me too, and my bus on Sundays went like 5 times to Freiburg. I hope you had it a bit better. But then Christian moved on to bigger places during his biochemistry studies. For example, Ulm, Mannheim, München, London, Canada, Massachusetts, and in the end to beautiful Basel. As you can already see from his biography, he has problems staying still and in the same place, and he loves to optimize his life. So besides developing new drugs and diagnostics for parasitic diseases, he also does sports like running, weightlifting, and yoga. And also, Christian is a part of our Science Slam Club in Basel, which means he's not only an active participant on this stage tonight, but he's also incredibly helpful in putting on the feet such events like tonight. So thank you for being in both roles and being with us tonight, Christian.
you, and me, we are all getting older. I especially feel it when I look in the mirror and I see my receding hairline. But thanks to my missing beard, soon enough, I look like a bald adolescent, like this guy here. But despite cosmetic changes, aging often brings serious diseases, with Parkinson's affecting 2% of the Swiss population. One of the key reasons for Parkinson is missing dopamine inside the brain. And so some researchers are trying to solve this with implementing things in your brain. Again, like this guy here. Nature already came up with something way more innovative. So let me introduce to you the Lancet liver fluke. The whole story starts with this guy here, Clark Ant. And Clark is a happy, hardworking ant. One day, he stumbles upon something jelly. He can't believe it. This is snail vomit. He shouts, this is some serious gourmet shit, and devours it all out of sudden. Little does Clark know that this meal will change his life forever. Suddenly, Clark starts to behave differently and has a certain urge to climb up grass blades, bite into the grass blade, and stays there. And especially strong is this urge during dawn and dusk. So one morning, when Clark climbs up the blade, bites into it, a hungry sheep comes along, sees the wonderful green grass, and thinks, this is a super meal, and eats both, the grass and consequently Clark. So what happened here? It all started with the infected snail vomit, which harbors the parasite, the Lancet liver fluke. And its sole purpose is to get into the sheep. And so once inside Clark, the parasite crawls up to the brain and hijacks the dopaminic receptors, making Clark in ways that it's more likely to get eaten by a sheep, like climbing up a grass blade, and even more fascinating, only at dusk and dawn, because this is the time when sheep graze. And you think now this little fluke is kind of a horror movie villain brain control? There's now actually the interesting twist, because this fluke is exactly what Parkinson patients could need. They are essentially devices that can highly sensitive control dopamine levels inside the brain. And so it could hold the key to new Parkinson treatments, making this fluke a superhero for us all. Thank you so much.
Super fascinating. So you mentioned very briefly Parkinson. So how developed is this strategy using parasites?
So far, the more interesting part to focus is not on the parasite, like I guess we will never eat just snail vomit and try to cure Parkinson. It's rather the mechanism that they have, and this is currently investigated because they can highly precisely target these different dopamine neurons, which is so far barely possible. And so rather than the products they have as little molecules, or even better, if there would be enzymes that could do this, that is would be the real breakthrough in this area.
I mean, I love parasites like viruses and everything that is so small and so powerful and can change behavior. Do you know of any parasites or diseases in the sense that change human behavior?
There are, we can start with rabies, which make the human more aggressive. That would be one thing, but the more fascinating changes would be actually the mice and the Toxoplasma gondii, also a great brain parasite, that makes that the mice is not afraid anymore of the cat, so it wants to battle the cat actually, and the cat eats it. But these are the closest examples we have.
Thanks. It really sounds like there's some, a really interesting and fascinating opportunities in this kind of mind mechanics that you were or the parasite mechanics that you were describing. So you talked about Parkinson's. What other sort of areas could this kind of stuff be helpful for?
This one?
Yeah.
So there's currently speculating, but the main jackpot Nobel laureate discovery would be if this would be modifiable enzymes that actually control the brain, because there are a lot of neurodegenerative diseases primarily that where you can't really target all these different receptors, also like the mental health crisis. And if we found something that could not only target dopamine receptors, but then also modify it to target serotonin receptors to make a multi-purpose tool like CRISPR-Cas before, that would be the real breakthrough.
Thank you so much, all the participants of the evening. And now this break is kind of an active break for the jury, of course, who need now to evaluate and make this difficult decision of the winner, the runner-up, but also for you, because we're going to see, I think, a QR code soon, is that right? That you can use to vote for your favorite participant or participants. Also, this is true for everybody who watches the live stream. One more thing, I don't know exactly how this time this voting is set up, but should this QR code work more than once for you, please contribute to make this a fair contest and don't use it more than once. Okay, we see each other back here in this room in 20 minutes, is that right? 15 to 20 minutes, so this is the time you have to find out the winner of today. Thank you everybody for watching. So everybody, the most exciting part of the evening. We will announce the winners. Please find back to your seats. Wow, that... So first of all, I would like to thank all the participants of tonight. You all did a great job, and everybody who voted discussed that it is so difficult and I'm sure it was the same for the jury. So a big applause, please, for everybody who participated today. There is several winners in a sense today. The jury themselves, they will now say who won and who is the follow-up. I will then later announce the favorite of the audience, and I give the word to the jury. I'm excited.
Yeah. On the stage. It's scary up here. Luckily, we don't have to present anything like you did, and really we're applauding you for this amazing performance. We learned so much. It was so fun, and it was really difficult to decide on a winner. We would have liked to hand out 10 certificates. So this was now our hard job, deciding on who did it best out of so many amazing performances. And in addition to those 3 C's that you've seen as criteria, we really thought about, did you manage to introduce the key aspects that you need to know to understand why your topic and the content is so important and amazing? And also, did you get to the key point of it? Did you understand the thing that makes it so amazing? And did you transport this to the audience? And now I've talked for much too long just to, get you more excited than you already are, and I'm handing over for the first result.
Wow, so very scary. So are you ready to hear who is the runner-up? So the second place is for Julia Hütte. Congratulations, Julia. Really, for an amazing, we learned a lot about dating, and it was great advice. We thought you did an amazing job. It was a very entertaining, really fun, presentation. It was explained scientifically very nicely, but really it was a lot of fun. So really, thank you for giving us really a lot of fun tonight.
Thank you. And now I've got the honor to announce the winner this evening. And as I'd like to say, as my colleagues have said, everybody did a fantastic job. Really, really great evening. So congratulations to everybody. It was really, really good. The winner is Sivaniya. So, Sivaniya, we were really impressed. The content was very, very clear. It was a very important topic that you were talking about and discussing. It was understandable. There was charisma. The use of the tools that you used to demonstrate your argument were very clear and understandable. So congratulations and well done and good luck. Thanks.
Our two winners, please stay on the stage. I'm going to ask other people to join the stage as well, but for now, we want to announce the favorite performance from the audience. And there were quite some votings. I saw the numbers was really impressive, and it was really a head-on-head on between two candidates. The winner who made it in the end is not yet on the stage, so somebody of the rest of the participants, and it is Shantanu. Congrats. Well done. And also for photo reasons and everything else, I'd like to ask all participants of the evening to join us on the stage now. And also the main organizer is our Sylvie, the person of the night. Please also join us if you can. If you have your photo duties, then you do that. Also, we have some helpers from the Science Slam Club in Basel. Jana, I don't know if you have time to come to the stage, but at least later. But first, our participants, please join us. Yeah, please come in the middle of the stage. And also, of course, a very big thanks goes to our jury who stayed concentrated the whole evening and did their very best to evaluate the best performances out of all the amazing performances. I think there is going to be a little present. Do you want a micro?
So it's really very local from my neighborhood in Zurich. Enjoy. Just something to enjoy and something for a great host and moderator as well.
Thank you. Thank you.
Thank you all for being here tonight. Save the date for the international final. I think we have the date somewhere on one of the slides. It will be a live event streamed also, so I'm really looking forward to seeing you there. Enjoy, and yeah, we're looking forward, and I hope to see you next year, either as part of organizing teams, jury members, moderators, audience, and looking forward to seeing you start to shine in science communication.
And thanks a lot to the audience. Without you, this wouldn't make sense, of course. So thanks for being here and spending the evening with us.
Maybe one with all of us who are involved. If that, whatever works.
Shall we do all first because everybody's on the stage and then we leave? Yeah. Jana, du auch. Komm.
Thank you.
Thanks.